Welcome, everyone. It is an absolute pleasure to introduce our speaker this afternoon, who is going to be part of our Distinguished Frankie Speaker Series. And our guest today is no one other than Carl Zimmer, most recently the author of the book Life's Edge, The Search for What It Means to Be Alive. But before I tell you more about Carl and the many books that he has written and the many ways in which his writing in the New York Times over the past 2 years has enlightened, informed, and taught us about the COVID epidemic, how it's unfolding, what we need to do to protect ourselves, and the evolving scientific understanding of the pandemic. Let me start out first by saying a few thank yous. I'd first like to recognize our benefactors, Mr. and Mrs. Richard and Barbara Frankie, who have been ardent supporters of interdisciplinary activities at Yale beyond the Frankie program in science and the humanities. We are deeply grateful to them for their support of all the exciting ventures that the Frankie program has undertaken over since its inception. And I'm also delighted to report that they are in attendance today. So welcome, Richard and Barbara. I also want to take this opportunity to thank Thai Camp for helping organize this talk and all the series that we have been holding so far, and Guy Ortoleva for his help with recording and making these recordings promptly available and accessible to many people beyond those who can join us today. So we are recording this event, so I would like to request all participants to leave your videos muted for the duration of the talk. Please go ahead and submit your questions at any time in the Q&A feature on the chat, and we will present these questions to Carl at the end. So now, let me move on to telling you a little bit more about our speaker today. So Carl, as you all know, reports from the frontiers of biology, where scientists are expanding our understanding of life. And I mean life in the fullest possible sense. And the New York Times has called him the country's most respected science journalist. And many of you can also attest to the fact, as I mentioned before, that in the past 2 years, what he has done has been invaluable in helping us stay on top of COVID. He's been reporting for the New York Times since 2004, where he now writes his wonderful weekly column called Matter. And his writing has won many, many awards, including the Stephen Jay Gould Prize, which is awarded by the Society for the Study of Evolution, in recognition for individuals whose sustained efforts have advanced public understanding of evolutionary science. In 2021, he was a member of the team at the New York Times that won the Pulitzer Prize in Public Service for coverage of the COVID-19 pandemic. Of course, in addition to his reporting, Carl is the author of 14 books about science. He's just incredible. He's such a remarkable writer with his insight, with his clear explanations. I mean, it's an absolute joy to read his books. And I have to gush here that the first book of his that I actually read was the one on viruses, which of course I reread during the pandemic. His latest book is Life's Edge, The Search of What It Means to Be Alive. And it's a notable book as picked by the New York Times for 2021. It was a finalist for the 2021 Penn E.O. Wilson Literary Science Writing Award. And so a little bit of his path, he started his journalism career at Discover, where he went on to work for 5 years as a senior editor. He's written for many other magazines, including the National Geographic, Wired, The Atlantic. And in 2003, he launched The Loom, an award-winning blog, which has been hosted by Discover and National Geographic. In 2015, Carl became a contributing national correspondent for STAT, a publication about health and medicine, where he hosted Science Happens, a video series that was a finalist for a National Magazine Award. In 2019, Carl Zimmer created What is Life, an 8-episode series of life conversations with leading thinkers about why life exists, how it began, and other big questions about existence. He's a 3-time award winner of the American Association for the Advancement of Science Journalism Award, twice for his work on the New York Times, once for the Loom. He's won the National Academy Science Communication Award. So the list of awards just goes on, and I will sort of briefly truncate it and just move on to tell you a little bit more about his books. So in 1998, he published his first book, At the Water's Edge, Fish with Fingers, Whales with Legs, and How Life Came Ashore and Then Went Back to Sea. And since then, he has written these 13 more books for which he has, once again, won fellowships from the Guggenheim Foundation and the Sloan Foundation. In 2018, he published She Has Her Mother's Laugh, The Power, Perversions, and Potential of Heredity. The Guardian named it the best science book of 2018, and the New York Times Book Review named it a notable book of the year. He's also won the 2019 Communications Award from the National Academies of Science, Engineering, and Medicine, and Science and Society Journalism Award. Amongst his other exciting books, the one that I have particularly loved is The Tangled Bank, An Introduction to Evolution, which was the first textbook about evolution ever published, intended for non-majors. And it was named an academic title of the year. One of the reasons I'm sort of going through a map of his writing is to show you his range, depth, and versatility in terms of the kinds of topics and the genres in which he has communicated so effectively with various sectors of the public. He also co-authored Evolution, Making Sense of Life, which was now a textbook for biology majors. And it was co-written with the University of Montana biologist Doug Emlen. And this is a textbook that is continually being updated with new editions. And I think the 4th edition is now in preparation. Carl is a popular speaker at universities, medical schools, museums, and festivals, as you can probably imagine, high demand, and has also, you've probably also heard him on Radiolab and This American Life. So he is the only writer after whom both a species of tapeworm and an asteroid have been named. So without further ado, as you can see, I'm very gushy about Carl's writing. I would like to turn it over to Carl. Carl, we're delighted to have you here today.
Thank you so much, Priya. It's really great to be here. Thanks for having me. It's an honor to be part of the Frankie Lecture Series. I wanted to talk today about life. I mean, as you had indicated in your introduction, life is kind of as a journalist, is my beat, as it were. But I wanted to sort of talk about life in the big picture rather than just one particular fascinating aspect of it. So let me just share my screen. I also wanted to thank the Frankie for this poster, which is just gorgeous. It's, sometimes you look at a poster and you're like, oh, they picked some weird photograph just to catch your eye. But this actually has a lot of meaning for me and a lot of relevance to my most recent book, Life's Edge, because this thing that you're looking at here, this is not out of some science fiction movie. This could be out of your backyard. This is an animal called a tardigrade. And here it's being photographed on, it looks like a bed of moss, probably, maybe grazing on a little algae. Tardigrades are incredibly common animals, but very weird animals, and weird in a way that's actually helps us to really think about what we mean when we talk about something being alive, like a tardigrade. So tardigrades, they're not visible to the naked eye. They're very small. You need at least a crude microscope to see them. And so people didn't know about that we're all surrounded by tardigrades until the 1770s, actually. There was a German preacher who named Johann August Goes. I'm probably not pronouncing that correctly. My German is not great. Anyway, so Goes was a preacher who, in his 40s, discovered natural history and just jumped into it with a huge passion. He actually collected tapeworms, and he had such a fantastic collection that the Emperor of Prussia bought it from him. I didn't know that Prussian emperors had an interest in tapeworm collections, but this one did. In any case, he also saw at a friend's house a microscope and was immediately taken with it. Microscopes had been in use for a century at this point, but people were still using it to discover new things. And he got himself a microscope. This is actually his microscope, and started looking at stuff. A lot of what he would look at would just be, water from a pond in his backyard and things like that. And he really marveled at this. He said, "A glass lens shows us a new world, and I know from experience that you will be full of amazement when seeing that for the first time." He was really quite taken with what he was seeing. And one of the things that really impressed him was this. This is his own drawing of this creature, which we call a tardigrade. Tardigrade is from the French. It means slow walker, but actually Goes didn't call it that. He called it a water bear. He said, "Strange is this little creature, because the whole organization of his body is extraordinary and strange." And he said that it at first sight has the closest similarity to a little bear. This also led me to give him the name, Little Water Bear. And then later he says, "One can hardly imagine something more horrible than our little creature appearing in the size of a real bear." That would be quite something. But in this world, water bears are quite tiny. And so Goes was observing it through his microscope, and he was struck by these 8 little feet that it has, which are each armed with 3 claws, and it was moving around under the microscope. He wrote, "His movements I observed were always the same. He was on its back, continually stretching out and retracting his feet as if he wanted to try to grasp something and to get on his feet." So here you have a naturalist who's observing a creature in action, the classic thing that naturalists do with life. So now, it's about 250 years later, and we know a lot more about tardigrades. We know that they are not just in ponds, not just on moss. There are tardigrades in the ocean. There are tardigrades all over the place, and they're an incredibly diverse group of species. And we can actually watch them walk, not just be on their back, flailing their legs around, but this is actually one of the first movies of a walking tardigrade. And they're not that slow for their size. They move pretty quickly on those 8 legs. So they clamber around. They are animals that move, and they move towards things. Usually they're moving to eat. They're finding something to eat, whether it's algae or some single-celled thing. And as tiny as tardigrades may be, they have a remarkable internal anatomy. They're surprisingly a lot like us, as bizarre as they might look on the outside. Sure, they have 8 limbs instead of our 4, but they have a brain and they have a mouth and they have a nervous system and they have muscles. They even have salivary glands and so on. You can see they have reproductive organs as well. So here's yet another feature that shows that they are like us, living things. And, living things, as a rule, reproduce. This is actually a slightly scandalous video of water bears having sex. This is how they do it. The male transfers sperm to the female, and then after fertilization, the female produces eggs, which it then lays, and then all those little eggs hatch, and now you have more tardigrades. So, I think we would all feel very comfortable to say that tardigrades are alive. I doubt that there would be many people that would dispute this. It seems obvious. I mean, we don't even have to discuss what we mean when we say something's alive. And yet, tardigrades can do some remarkable things that actually challenge our comfortable assumptions about life. So if you were to become dehydrated, let's say you lost 5% of the water in your body, you would feel terrible. You lost 10%, you would feel worse. If you start losing 20% and more, you're going to die. That's it. You can't survive. And if I were to pour water on you after that, you would not suddenly sit up and start talking. You're gone. You've died from dehydration. That's not true for the tardigrade. So the dehydration, desiccation, drying out, that's a regular part of being a tardigrade. It's just that they're in environments often where they are going to get dried out. And when that happens, they don't just become sort of a shriveled version of themselves. They go through a very remarkable change. So this is from an article in American Scientist from a couple of years ago. So on the top, it shows a regular water bear. Below it is a water bear that has been dehydrated. And this strange little shape, it's called a tun, T-U-N. And there's nothing going on in there. Tardigrades have to breathe. These don't breathe. Tardigrades have to eat. These tuns do not eat. They do not excrete waste. They do not move. They look like a grain of sand. And yet, if you were to pour water on that tun, it would swell up and it would go back to looking like the tardigrade at the top there. And you could wait quite a while to do this. You could wait decades. And scientists they have stored away tons in collections. And eventually people have said, here's a tardigrade ton that's been here for 30 years or 50 years. Let's see what happens if we put it in water. And more often than not, they're able to come back. And they really look very much like just an ordinary tardigrade, just walking around, eating, just in a sense, being alive. And what's incredible is that this capacity, it makes tardigrades unbelievably tough, resistant. They can be blasted with levels of radiation that would kill any other animal many times over. They've been sent into space, actually, in this little craft that was shot into orbit. And the tardigrades that were sent up there, they were actually exposed to just the vacuum of space. And they rode around like that in outer space, no spacesuit, and then came back to Earth. And then scientists were able to revive them again. So these tardigrades are one of the most extraordinary creatures on Earth. And so scientists are wondering, well, how do they do this? How is this possible? It's a very complex process that they've evolved. So the first step is actually to to avoid the damage that comes with dehydration. So the problem with getting dehydrated is that all of our proteins and our DNA, they really need to be in water to work properly. And so once the water levels start dropping down, proteins can get misfolded. There can be molecules that clump together, and things just stop working. So tardigrades do this amazing thing where they make a huge amount of a special sugar called trehalose, and they basically replace the water in their cells with trehalose, and they can kind of shrink their cells down. So this is part of the reason why a tardigrade becomes really tiny when it becomes a ton. But that's just the start, actually. So they then, this diagram, which is really complicated, is just reflects all the things that are happening inside of this animal's body as it turns into a ton. You see a little line there, vitrification. Vitrification, you might think of like glass. And that's kind of what happens, actually, that it makes a bunch of proteins that wrap around DNA, mitochondria, all sorts of different structures, and freeze things in place and protect them from damage. So essentially, the tardigrade is becoming a kind of a protein glass. And, if we think of metabolism, for example, as being like essential for life, these tardigrades aren't doing it. Their proteins cannot move. They cannot react with each other. That's it. And they can stay like that in this state for decades. So is the tardigrade that's become a ton, is it alive? Well, if we turn to dictionaries for some help, it's a little difficult. So you have a definition here. The first one, the quality that distinguishes a vital and functional being from a dead body. Well, what's that? I mean, it's almost kind of circular. You're saying that life is something that living things have. Okay. The second one is a principle or force that is considered to underlie the distinctive nature of animate beings. What is that? This principle? It sounds almost like alchemy. C is a little more concrete. An organismic state characterized by capacity for metabolism, growth, reaction to stimuli, and reproduction. Okay, I mean, we can get our heads around that scientifically, but does a tardigrade ton metabolize? Nope. Can't eat, can't produce waste, doesn't do those reactions. They just do not happen. There's no water there to allow them to happen. Can it grow? Nope. Perfectly fixed in size. The cells do not divide because they can't do metabolism. Do they react to stimuli? Well, with the exception of adding water, no. They just it's essentially like a grain of sand. Can they reproduce? Absolutely not. So we're left here with a very frustrating situation where we have something that in its ordinary form we would agree is alive, is life, but can go into another state, a state that really does not fit our definition of life. I'm sort of reminded of all the terrors that humans have had about being buried prematurely. Because people are worried that they might go into some sort of deep sleep or some type of state that would be mistaken for death. But in the case of tardigrades, it's very similar in some ways to death. Scientists don't call it death; they call it cryptobiosis, which means hidden life. So cryptobiosis, which other animals can do as well, it is in a way sort of taking life and sort of tucking it away in a safe place. But the fact is that by a lot of the sort of standard ways that we talk about life, a tardigrade can stop being alive and then be alive again. There's another example that's it's also really fascinating in a puzzling way, and that's viruses. This is a reconstruction of the SARS-CoV-2 virus, which causes COVID-19. So if like me, you've had COVID, you had a billion of these things inside of you, replicating away. Tardigrades are small, viruses are much, much smaller. But obviously, size doesn't really take away from the power that they can have. I mean, this little virus has turned our whole world upside down, has wrecked global economies, probably killed about 18 million people so far worldwide. And aside from the very practical specifics of what viruses do to our lives, they're really very puzzling in and of themselves. Like, what are these things? Are they alive? We couldn't even begin to ask these questions until people had found viruses. Because viruses are so much smaller than tardigrades, it took even longer for people to be aware that we're surrounded with them, that they're inside of us. So tardigrades were found in the 1770s. Viruses really weren't recognized until for about a century later, a little over a century later. Obviously, there were diseases like rabies and influenza that made people sick, but people couldn't say that's a virus that's causing it. That's something that belongs to this class we're going to call viruses. It actually wasn't a human virus that was the first one that was discovered. It was actually a plant virus. Tobacco farmers in Europe were dealing with a disease called tobacco mosaic disease. And it would cause these splotches that gave it its name, kind of like a mosaic. And so some scientists like Martinez Bayerink figured out how to do an experiment to understand what was causing the disease. They thought, well, maybe it's a fungus or bacteria, because by the late 1800s, the germ theory of disease was becoming pretty well established. So what they did was they would mash up a sick tobacco leaf and mix it with water, and then pass it through a filter, so that that big clumps of stuff couldn't get through. Only small things could get through. And they would inject that fluid into a healthy plant, and then they would see that actually that plant got sick as well. So that suggested, yes, there was some kind of germ that was causing this disease. So then Bayerink actually used another filter, a very, very fine filter, one that was where the pores were so small that bacteria and fungi could not get through. So he did the same experiment again, mashed up a leaf, mixed it with water, passed it through a filter. Now he's got something that really just looks like water. And he takes that and he injects it into a leaf, and lo and behold, the leaf gets sick. So he's saying, well, there's something else out there, something other than bacteria and fungi. I'm going to call it a virus. So this is the first time the word virus is used to describe these invisible agents of disease. Couldn't see them. It's not like he was looking through a microscope at his lovely little water bears. He could see the effects of the viruses, but they were too small for him to see. This remained the case for decades. The viruses remained invisible. Scientists were now able, though, to conceptually understand viruses and could then look for human viruses, like, for example, influenza. And so still, though, the microscopes of the early 1900s, while they were much better than the ones that Goes had in the 1700s, they still just weren't powerful enough to see a virus. So Wendell Stanley had this interesting idea. He knew that you could make crystals out of different substances, we're familiar with salt crystals, but you can actually make crystals out of proteins too. They, under the right conditions, they'll come together in this sort of some repeating pattern. And if a crystal gets big enough, you can see it. You can shoot X-rays at it and figure out its structure. So I thought, well, there's some indication that viruses have protein in them. So why don't I make a virus crystal? So he grows a lot of tobacco mosaic virus in tobacco plants, concentrates them, and then he sees, lo and behold, that these little spiky shapes start to form. They start to become big enough that he can see them with the naked eye. And he can collect these and store them just, on a shelf in a jar. And it's kind of like cryptobiosis. He can take them off the shelf months later and, put them in water and let them infect a tobacco plant, and they cause just as much disease as before. So, so this idea that something that seems like it's alive can be a crystal, like salt, this is kind of mind-blowing. And when Stanley published his results, the journalists reporting on this really had a hard time keeping their composure. So the New York Times, they would talk about this, how this staggers the imagination. The viruses are neither dead nor alive, kind of like a tardigrade ton. And they seem to lie on that shadowy borderland where matter is ready to leap into life, which is a line I just love. The subject really just for years kept obsessing people. And actually in 1940, a few years later, there was another article which had a section that was headed, "Can Crystals Live?" And they talked about Stanley's experiment, and they're asking, were these crystals alive? Apparently no more so than diamonds, glass, sand, or other crystals with which we are familiar. Yet when virus crystals were put on a tobacco leaf, the mosaic disease spread like a slow fire over a whole field, just as if it had been infected by living bacteria. So, so very early on, even before we really knew very much at all about viruses, people recognized that they pose a huge challenge to our concept of life. So finally, microscopes caught up with the viruses. So electron microscopes made it possible to actually visualize viruses for the first time. This is what tobacco mosaic viruses look like. They have this interesting cylindrical shape. What's inside of them? What are these cylinders made of? Well, Rosalind Franklin, who might be best known for her work on the structure of DNA, before that had actually made a name for herself by studying tobacco mosaic viruses and actually doing really important work to figure out, what are these things made of? What are viruses made of? And it turns out that that a tobacco mosaic virus is made of protein. Here, the blue and violet show different proteins that are kind of locked together. And they hold genetic material. In the case of tobacco mosaic viruses, that material is RNA. We keep our genes in DNA, which is a double strand. There's some viruses that also have double-stranded DNA. But a lot of viruses have RNA for genes instead. That's one strand. So COVID-19 is caused by an RNA virus. The flu is caused by an RNA virus. Tobacco mosaic disease is also caused by an RNA virus. But what's really interesting here is that it's just, this protein is just a package. And it's just carrying these genes. It doesn't even contain enzymes that you need to copy the genes like we have. It's just a delivery system. Now, it turns out that shape of tobacco mosaic virus is not the rule for viruses. And as you can see here in this diagram, you have viruses that are like soccer balls. Some are like snakes. Some look like weird little spaceships. They're all very small, but they have this incredible variety. There could be literally trillions of species of viruses on Earth. We don't know. We barely scratched the surface when it comes to their diversity. But in spite of all the diversity, there are a lot of things that unite them. In some ways, if you've seen one virus, you've seen them all. Again, in the sense that the viruses are not like cells. So they're not little chambers full of metabolism going on inside. A cell can grow, a cell can divide, a cell can copy its own DNA. Viruses can't do any of that. They're a protein shell with genetic material inside. And Sir Peter Medawar, a famous immunologist of the 20th century, said that they were simply a piece of bad news wrapped up in a protein. That's a good way of summing up what a virus is. So what this piece of bad news does is infect cells and get the cells to make more bad news. In other words, the protein shell will have receptors on its surface that can let it latch onto a cell and deliver its genes inside the cell. And the cell's own machinery reads those genes, makes proteins, and those viral proteins basically take over the cell and cause the cell to make new viruses, which then escape the cell to go infect other cells. So just with the real basics there of what a virus is, we have the question again, as we had with tardigrades and their ton, is a virus alive? Now, I showed you the dictionary definition before. Scientists actually will come up with their own definitions of life. And I would say that the most influential is one that a group of scientists came up with for NASA. Sorry about that. In the early 90s. They were having a meeting about exobiology, about finding life on other planets, and they were like, wait, we need to agree on what we're looking for, right? And so they had spent all day arguing over this, argued over dinner, and by the end of the evening, they had settled on a working definition. I write about this whole process in my book, Life's Edge. I said, life is a self-sustained chemical system capable of undergoing Darwinian evolution. So short, sweet, clear, good writing. And as I say, very influential. Like you will see this definition or versions of it around a lot. So we have these viruses. Darwinian evolution, that's a big part of the definition. They certainly are. We're dealing with the Omicron variant of SARS-CoV-2 right now. Variants are produced through evolution, through mutations, through natural selection. So now, Omicron is far more fit than the first viruses that emerged in China. It has evolved a lot since it became a human pathogen. So no question that viruses are capable of Darwinian evolution. They're the textbook case of it. However, what about being a self-sustained chemical system? Well, there are chemicals in it, obviously, but self-sustained, in the sense of like being able to feed itself, to expel waste, to maintain an internal environment. Nope, can't do it. It just doesn't do it. So here we have something that straddles the definition, which meets the definition halfway. Is it half alive? Can we have such a thing? Gerald Joyce is a scientist who helped to come up with this definition and wrote about how they came up with the definition later. And a reporter was asking him about that experience and brought up viruses. And Joyce said to the reporter, according to the working definition, a virus doesn't make the cut. So sorry, viruses, they're not alive. Now, when I talk about this in my book or in lectures, scientists sometimes will reach out to me and they'll say, well, of course, Joyce is right. I mean, viruses are not alive. I mean, they don't have those essential things that make life possible. And then I will also hear from other scientists who say, well, this is ridiculous. Of course, viruses are alive. You can't understand them without thinking about them as evolving living things. I literally got 2 emails in the same day where I was being told that viruses were and were not alive from scientists. So this is still a live wire issue. I think one of the most intriguing ways of addressing this and challenging Joyce comes from someone named Patrick Forterre. He's a French virologist, and you could kind of think of viruses, according to Forterre, a little like the tardigrades, in the sense that like tardigrades can be in 2 different states. So you've got the regular tardigrade walking around, and then you've got that ton. So a virus that's floating around outside of a cell, think of that as kind of like a ton. It's got, genetic material inside and so on, but we wouldn't really, not really alive. But once it gets inside of a cell, things are very different. It's not so much the virus itself that's alive, but it is the merger of the virus and the cell. So an ordinary cell, Forterre calls it a ribocell, and that is because they have ribosomes, which are these factories that they use to make proteins based on their genes. But the cell is making proteins for its own needs. If a cell gets hot, it can make proteins that will help to protect itself against heat. It can make, proteins to stick on its surface to grab different kinds of things that it needs to survive. So a cell, can look out for itself, as it were. Now, once a virus gets into a cell, as I mentioned before, all of a sudden there are all these virus proteins inside of it. And these viral proteins completely change what the cell does. In the case of COVID, for example, if you get COVID, the cells that are infected with SARS-CoV-2 inside your body, they can no longer make their own proteins. The virus proteins plug up those ribosomes, and they only let virus genes in. Virus RNA can go into the ribosomes. So it's like the worst, bouncer at the club door. It's not going to let in our RNA. It's only going to let in the viral RNA. And so in other words, the cell's purpose, the cell's function has changed, and he calls it a virocell. And in that regard, Forterre says viruses are alive. And it's easy to sort of to miss just how much a virus changes a cell once it gets inside. So I mentioned it plugging up those ribosomes. Here, the ribosomes are shown in purple in this computer image of an infected cell. But these yellow things, these are actually represent bubbles that the virus creates inside of an infected cell. So the cell is full of these bubbles, and inside of these bubbles gets put new viral RNA. In other words, genetic material for new viruses. It builds up inside of these little nurseries, and then it will later then get packaged into new viruses to leave. So again, like if you look inside of an infected cell, a virocell, it just doesn't look like an ordinary cell. It looks like a different kind of life. And so Forterre, who has a knack for the poetic, says, whereas the dream of a normal cell is to produce more cells, the dream of a virus cell is to produce 100 or more new virus cells. Now, I don't know when this debate's going to end, and I'm not sure maybe it can end in the way that people are conducting it. And to sort of explain what I mean, I want to just jump back again to the 1930s, back when viruses were a deep, deep mystery. Norman Peary, who was a British virologist who did a lot of important work on tobacco mosaic virus and other viruses, he wrote about this exact same question. He said, when one is asked whether a filter passing virus is living or dead, the only sensible answer is, I don't know. We know a number of things that it will do and a number of things that it won't. And if someone will define the word living, I will try to see how the virus fits into the definition. So Peary sort of recognizes that we are trying to pretend that there's a clear scientific question to be addressed without recognizing that the language that we're using to frame the question is actually really problematic in some ways. If someone will define what living means to me, well, gee, I gave you the NASA definition of life. I gave you the Merriam-Webster definition, but there are lots of other definitions. These are actually like taken from the scientific literature. And these are just a tiny sampling. There are several hundred published definitions of life, different ones, and more get published every year. So if Norman Peary was waiting for someone to give him a very clear definition of life that he could work with to figure out if viruses are alive or not, well, 80, 90 years later, we're still waiting for that kind of clarity. And maybe that this buzzing confusion of definitions tells us something. That's the argument that Carol Cleland, a philosopher at the University of Colorado, has made. And she wrote a book a couple years ago called The Quest for a Universal Theory of Life, where she lays this out. She lays it out as a philosopher. And the key point that she makes is that we should not be searching for a definition of life. In fact, definitions of life get in the way of our understanding of life. And that might seem weird. I mean, how can you understand, study something that you can't even define? That sounds like mystical, but it's actually not. The problem is that definitions, philosophically speaking, are ways of organizing concepts. And Cleland points out that, let's say, what is a bachelor? You can define a bachelor because all you're doing is you're relating different concepts to each other. So a bachelor is an unmarried man. Well, what does it mean to be, that there's no question about that. Now, what is marriage? Well, that's complicated. What is a man? That's complicated too. And so, there are things that can be easily defined, cleanly defined, and things that are much more problematic. And if you focus on trying to find just the perfect definition, you may not be spending the time you need to really understand what it is that you're dealing with. So an example of that she gives is water. Okay, so what is water? How do you define water? Well, 500 years ago, if you went and talked with alchemists, they were the ones who understood what we call chemistry best, and they would define it for you as being transparent, as being liquid, as being able to dissolve certain things. they give you a list of characteristics, but that was very problematic because, for example, what happens when water gets cold and suddenly you have a solid? Is ice water? Well, no, not according to the definition. Then you would have these different kinds of transparent liquids that could dissolve things, but some could dissolve some things and some could dissolve other things. So are they all water? Alchemists would try to deal with this by calling some things royal water, for example, which we would know to be certain kinds of acids. Even Leonardo da Vinci at the time was just left mystified by water. So you would talk about how water was sometimes weak and sometimes strong, sometimes bitter, sometimes sweet, sometimes thick or thin, sometimes bringing hurt or pestilence, sometimes health-giving. So it suffers changes in as many natures as are the different places through which it passes. Leonardo da Vinci would not have gotten any greater clarity just trying to come up with a definition of life. What was needed was 300 years of research, of experiments, of thinking and theorizing, and coming up with a theory of matter in which you could explain water. So once you have the language of atoms, molecules, elements, and so on, you can think about water in terms of H2O. And you can think about water as being molecules in motion, and at certain temperatures, it becomes ice. So yes, ice is water. Sure, of course it is. And this is, it's not so much that you're seeking to define water now you're just trying to understand it. You're trying to see how water behaves in different ways and make predictions about it and so on, and use it in interesting ways that you hadn't expected before. So this is the difference between a definition of life and a theory of life. We don't have a theory of life, but what would it be like? Well, it would be a way to explain how matter takes on certain properties. And we could debate over what those properties are, but what I like is the analogy with superconductivity. Over a century ago, scientists discovered superconductivity. It was very weird. There were these substances that lost their resistance to electric current when you got them very cold. It didn't make sense. Einstein couldn't explain it. Richard Feynman couldn't explain it. But eventually, some folks got together and made a theory of superconductivity, a theory that explained it in terms of pairs of electrons. And now it made sense. Now you could see how different substances could actually be superconductive. And that then allowed people to start making new kinds of superconductive material. And you could, start making things that could support trains, for example. So it's a theory that we need, a theory perhaps about, organization and information. Maybe that's something that really is what needs to be explained for life. Just to give you one example, there's a theory called assembly theory that Lee Cronin at the University of Glasgow has been exploring. So it's not about the specifics of DNA and protein, but just about the steps that it takes to make molecules. So maybe what's distinctive about life is that life and only life can make certain molecules that require lots and lots of steps of assembly. That it's a simple idea, but it's actually potentially a very powerful one, because maybe you don't need to know in advance what living things are made of on other planets. You don't need to be just looking for DNA-based life. Maybe there's, on a moon like Enceladus over at Saturn, maybe life is totally different. Maybe assembly theory can help us to understand it. And, it's possible that, maybe life isn't going to be discovered on another planet. Maybe someone in a lab is going to make it. These are little droplets of mixtures of oil that actually Lee Cronin and his colleagues have produced. Just mixing different things around, you get droplets that do weird stuff. He refers to them as unstable protocells. They move around, they divide, They do life-like things. They're very strange. I don't know we quite call them life, but they might be telling us something about how living things first arose. And we could use theories to understand what we're looking at. But this really takes us back 250 years, because, when we're looking at these amazing experiments, like what's happening in this dish, I think we're kind of like Goes was, where he talks about a glass lens that shows us a new world. I think, that we, in a sense, are looking at life for the first time with this new perspective. And we're just beginning to be understanding it in this new and profound way. So thank you so much for your time. There's lots more about these issues in my book, Life's Edge. And I'm more than happy to take questions if people have them.
Thank you so much, Carl, for that wonderful talk outlining the complexities of the semantic definition of what life is, because of the biological, chemical, and other properties that we attribute to life forms. And this is a question that's of great importance even in the space sciences at the moment, where people are trying to understand if we do discover signs of extraterrestrial life, what might that look like? And there, of course, we may have to even expand beyond the idea of carbon-based life, which is where we are fighting over the definitions on Earth. Thank you so much for that wonderful talk. So let's give Carl a clap, a virtual clap, before we open up for questions. So I think we have a few questions that have come into the chat. The first one is from Shona, who would like to hear your thoughts on the theory of symbiogenesis, taking more of a center stage in common knowledge and the future role of quantum mechanics in explaining questions in biology.
Wow. Well, great questions. Yeah. So symbiogenesis is this idea of different organisms or different living systems coming together in a symbiosis to create new levels of life. So, for example, the best example would be in our cells. We produce fuel in mitochondria. And those mitochondria are basically our little, little house guests. They're bacteria that took up residence inside of our single-celled ancestors, maybe I guess about 2 billion years ago, something like that. And there was a mutual benefit there, in the sense that the bacteria were getting shelter, and they were getting some provisions, and then they could actually produce things like ATP, fuel for the host cell. And eventually it became impossible to distinguish the two of them. There became just one greater unit. When people like Lynn Margulis were arguing for this in the 60s, it was fairly controversial. Wasn't clear how important it was, but along with mitochondria, there are lots of other examples. Whether you're talking about the chloroplasts that make plants green, or insects. Insects and other invertebrates have lots of bacteria inside of them. The bacteria actually inside of their cells and can only live there. And they have this remarkable symbiosis. So like cicadas, for example, which, we have these emergence of cicadas, and the cicadas are eating fluid from trees, and there's not much nutrient in it. And on its own, the cicada couldn't survive on it. But you feed that to the bacteria that it harbors, and the bacteria can actually build essential amino acids and then give them to the insect. And so you have a symbiogenesis right there as well. I don't think that symbiogenesis will necessarily tell us that much about the very origin of life, just because you need those complicated things to begin with to have them to come together. Bacteria are already pretty complicated. But it is possible to think about, people think on the early Earth, the earliest life forms might have had RNA inside of them, or maybe just RNA floating around in sort of oily layers at the bottom of ponds or something like that. So you could have different RNA molecules cooperating with each other in a kind of molecular symbiosis. That's possible. As for quantum mechanics, I mean, obviously, like when you get down to the level of atoms, quantum mechanics matters. I don't know. I mean, ever since when I started as a science writer over 30 years ago, people were talking about the revolution of quantum mechanics to change the way we think about biology. It's important, if you're understanding like vision and, for example, and levels of energy levels of photons and so on. But I don't think that I haven't seen much yet in how quantum mechanics really changes the way we think about important kinds of biology or principles of evolution. But we'll see. That could change.
So somewhat related to that, Carl, I had a question. So, I mean, in our view of evolution, right, it's very much driven by this idea of competition. And so, as you just mentioned, right, so isn't there sort of an emerging understanding now on competition and cooperation, both being elements of how evolution actually operates?
Sure, and Darwin was intrigued by cooperation from the very start of evolutionary theory. I mean, he certainly was aware of the importance of cooperation. I think that basically, there are situations in which cooperation is going to be the most advantageous evolutionary strategy. And there are going to be situations, and for certain organisms, where it's not. When SARS-CoV-2 is infecting us, for example, and you get mutant variants popping up inside your body, there is a competition going on among those viruses. It's an unconscious competition. It's not like a race or something. It's just like the question is, well, which the viruses with different mutations, which ones will make the most viruses? And then over the long term, the mutations that increase the fitness of viruses the most may become the ones that are dominant. On the other hand, the fact is that viruses, this is kind of weird, but viruses do sometimes cooperate. They actually work together to different kinds of viruses will get into the same cell at the same time and manipulate it together using their pooled resources to make more of each kind of virus. So, that's, you're still getting sick. So that cooperation isn't very warm and fuzzy for you, the sick host, but it's pretty cool that the viruses are cooperating as well. So they're both definitely important for evolution.
Great, thank you. So we have a question now from Deepthi Pradhan, who runs the famous Tilda Science Cafe. And her question is, where would one place viruses on the tree of life?
Great question. Yeah. So the tree of life was this image that Darwin invoked in The Origin of Species, because he saw species as sharing a common ancestor, evolving, splitting off from a common ancestor like branches on a tree. And so we share an ancestor that lived maybe 6 or 7 million years ago with chimpanzees. We share an ancestor that maybe lived 10 million years ago with gorillas. Those are our closest living relatives. And, for animals, like you can make a whole tree, like you showing how animals are all related and descend from a common ancestor. There was a primal animal from whom we all descend, and that's all animals. That's tardigrades and jellyfish and sharks, all of us. And if you step back further, we share a common ancestor with fungi. And if you step back even further, we share a common ancestor with plants. So we're closer cousins to fungi than plants. It's something that's worth bearing in mind. Step back even further, and you can start to see how we're related to protozoans and then to bacteria. So all of cellular life can pretty well be organized on a tree of life, looking at their genes and comparing them. Then you got viruses. What do you do with these things? How are they related to, if at all, to anything else? Part of the problem is that viruses are so small and they have such tiny genomes that there's just not that much information to to link them to anything. So it's very hard to know. I mean, you can tell how viruses are related to each other, but it can be very challenging to link them to the rest of life. But there are some clues as to how, genetic material from cellular life may be able to break off and become viral. In fact, it goes the other way as well, that, we have been infected by viruses, our ancestors, for millions of years. Some of that viral DNA has been incorporated into our genome. So we're about 8% of our DNA comes from viruses. It's possible viruses were there at the very beginning of life. Maybe viruses started out as parasitic RNA. We don't know. So viruses may sort of like hover around the tree of life. Maybe that's the best way of thinking of it.
Thank you. I think you've definitely shown us the paradoxes of how we could deal with the viruses. So the next question is from Agatha. Which of the definitions of life resonates most closely with you, and why?
It sort of depends on the day. I read about the creator of one of those definitions in the book, named Albert Szent-Györgyi. He was a Hungarian Nobel Prize winner. He won the Nobel Prize for discovering vitamin C, basically. Once he had won the Nobel Prize for vitamin C, then he decided, well, okay, now I can get down to some really serious work. And so he really wanted to get down to the very nuts and bolts of life, really like life itself on a sort of biophysical level. And he did amazing work. He was in Hungary and was starting this research right when World War II breaks out. He becomes a spy. He joins the resistance, and he's on the run around Hungary, but he will show up every now and then unexpectedly in his lab to make sure the experiments are going okay, where they're trying to understand how muscle fiber works, because he really feels that that's going to give the key. And then he actually then smuggles out. He gets a Swedish diplomat to smuggle out the manuscript about these findings, because he's terrified that no one's going to know about what they found. And a lot of people think maybe he should have gotten a second Nobel Prize for that, but he joked afterwards, after that work and after the war, that he was working so hard to figure out what life was, but it was just sort of, it kept escaping him. And after a while, he was just like, well, I call life a play of water, because he was interested in the way in which, a lot of the key reactions that happen in a cell involve water molecules, being broken up and being used to carry out these reactions. So he just said, life is just a play of water. So I can live with that.
So the other question, the next question is even from Madhusudan, who says even self-sustaining living systems cannot be a closed system. They need external sources of energy. If you put things in that way, is a virus that different? They need external matter to exhibit other signs of life. And they're asking for your comment.
Yeah, no, that's a very interesting point. You know, I think in biology, there's been a very long-running division between parasites and quote unquote free living hosts. And it's, you know, but you're right. Like, I mean, who is really like totally free living? Like, I mean, we depend on other living things. You know, we all carry around a microbiome. We'd be very sick without it. And so, so I agree that it's you know, we can't be too contemptuous, as it were, of viruses. But I still think that, you know, the fact remains that, you know, viruses, you know, are really different than cells in some fundamental ways. But who knows, you know, scientists are finding some really weird viruses these days, like really big viruses with lots and lots of genes. And some of those genes seem to encode for parts of cells, you know, like ribosomes or parts of the apparatus for photosynthesis. So there's some weird things about viruses. I'm sure we'll find viruses that really, you know, defy our easy definitions even more.
Thank you. Thank you. So there's a comment from Gary who says, you know, what is life? The question needs a context and who is asking and in what domain. And the important issue is knowing the mechanism as to what is going on in a particular domain. And then you can assign terms, either new ones or old ones, to different parts of the process to further experimental work.
Well, you know, certainly, you know, it helps to, it can be useful to have working definitions of life for particular work, you know, the working definition. So, you know, if you want to go and find, you know, alien life, and you're going to spend hundreds of billions of dollars trying to do it, you know, like it might be nice to be able to know what it is that you're even thinking about finding. But even then, you know, I talked to an astrobiologist who appears in the book, Lori Barge, and I said to her, you know, so she's Enceladus, which I showed before, this moon of Saturn. She'd really like to go to Enceladus because it's got it's got covered in ice, but it clearly has a lot of weird chemistry happening inside of its liquid ocean. Mm-hmm. And I'm like, well, what's your definition of life? Like, what would it have to be in order for you to say it's alive? And she's like, I prefer not to deal with things like, I don't like to define life. You know, this is literally an astrobiologist, you know, whose job it is to find life. She's like, you know, I'd like to go there and see what the chemistry is. And I bet it's very interesting. And I'll just want to understand what it's doing and what makes it interesting. And, you know, maybe eventually we might come to agree that there's something there we call life. But, you know, what if it was, you know, chemistry that in 10 million years will give rise to life? You know, like you wouldn't want to just dismiss it out of hand before. So it's interesting. Even astrobiologists sometimes don't want to define life.
Thank you, Carl. The next question is from Brian. Are there genetically similar creatures to tardigrades? If so, do they display the cryptobiological process?
So tardigrades are they're animals. They're you know, I forget what they're, scientists are always trying to figure out how closely different parts of the animal kingdom are related to each other. So I think they're kind of closely related to insects, as I recall now, but, you know, they're their own branch. But, you know, so they share a common ancestor with other animals like us that have brains and guts and nervous systems and so on. Now, cryptobiosis, what they're able to do is actually found in a number of animals. Lots of little invertebrates that do it. And fungi and bacteria seem to be doing something similar as well. So what's interesting is that, I showed you the tardigrade strategy, which is essentially to just replace your water with sugar and then turn your body into basically a protein glass. That's one way of doing it. Other branches of the tree of life have evolved other strategies to do the same thing. But the result is the same, that these things are somewhere between life and death and can stay that way for decades, as far as we can tell.
Great. The next question is from Diane. So since prions lack genetic material, is there consensus that they are not alive?
Again, it all comes down to what your definition of life is. And so, they are again kind of like they're at life's edge. They might fulfill some parts of common definitions of life and fail others. So for example, prions are proteins, just naked proteins, but they are sort of deformed versions of proteins that we make ourselves. And so if a prion version of the protein comes up next to a regular version, it can deform it as well. So now that we call that a prion, and then that can deform another protein and so on and so forth. So that can lead to diseases like mad cow disease that people have gotten from eating contaminated beef or lamb. Deer suffer from a prion called chronic wasting disease, which is a big problem. Deer hunters are aware of that. But again, it's just a protein that can it reproduce? Well, it doesn't make a new protein from scratch. Neither does a virus. All the SARS-CoV-2 in the world is made by us. It's made out of human molecules. And so, it's sort of weird that we think of this thing as a virus, but it's manufactured out of human parts. So I don't know. So is making another molecule take your shape? Is that what we call that reproduction? It's very hard to say, and yet it has some lifelike properties. So again, it's not really, the answer isn't, maybe we shouldn't be thinking sort of either or. I know we like to think of life or death, something's alive or it's not, but we have these very fuzzy cases that are interesting because they're so fuzzy.
Right. Thank you. So the next question is from Stephen, who asks, it seems that exceptions have been the most helpful in trying to establish a rule. How do the characteristics of mitochondria support or fail to support their candidacy for life, especially given their supposed origins?
Yeah, so I could have spent the hour talking about mitochondria instead of tardigrades or viruses, because here we have another fascinating example. Are mitochondria alive? Now, we'd probably all agree that their ancestors 2 billion years ago were alive, in the sense that other bacteria floating in the ocean are alive. They were able to metabolize and reproduce and evolve. What's kind of amazing is that actually inside our cells still, mitochondria divide. They're sort of like doing their own thing. The mitochondrial division and cell division are kind of tied together, but not totally. So, different cells have different numbers of mitochondria in them, which is interesting. So they're still doing part of what we think of as being alive. But the fact is that if you look under the hood, if you open up mitochondria and look inside, they still have some genes I forget the number, something like 30 genes. So they have some DNA inside of them. And those genes produce proteins that do incredibly important things. We get really sick if those genes are mutated. Those genes, descend from the genes that were carried by free-living mitochondria. But, those mitochondria had thousands of other genes. So where'd they go? Well, some of them were lost. They just didn't need them anymore. Some of them actually ended up getting moved from the mitochondria into our own chromosomes, into our own DNA. So we have some, and along with the viral DNA, we have some bacterial DNA in there as well. So you have this thing, like if you take mitochondria and pluck them out of a cell and just put them in a petri dish and say like, okay, on your own, like they're dead. They can't do it. And yet there's nothing more important to our own life than mitochondria. You shut mitochondria down, we're dead. Some of the most potent poisons work by shutting down mitochondria. So I have no idea if you call mitochondria alive or not. It's really up to you, but it's really interesting to think about how it compares to things that we're more comfortable calling alive.
Thanks, Carl. I think true to your book's title, right? There are many questions that one can ask about the edge of what counts as life and how we define that edge. But before we close this wonderful session, it is too tempting to let you go before asking you what you think is coming next with COVID, with our pandemic. Are we in for another minor wave, unnoticeable wave, noticeable wave?
Well, we're 2 years into this pandemic, and I find that the smartest scientists I talk to are also the most humble. They really respect this virus's capacity to surprise us. Nobody saw Omicron, for example, coming. Like no one had said like we're going to have something like Omicron until we had Omicron. So, people keep saying like, oh, that's it, we're done. Or, oh, virus has always evolved to get milder, or oh, the virus has run as far as it can go. And I honestly don't think that we know that or not. And that's not a happy thought, given all the suffering that it's caused us already. But right now, today, as we speak, there is this subvariant, a subvariant of Omicron called BA2, which has become dominant. Most cases in the United States are that, but cases are still pretty low in the US. And it's possible that this is going to give us a bump, but on the other hand, a lot of people have already been infected recently, or they've been vaccinated, so they may rein that in. I don't really think we know. I mean, we will know in a week or in 2 weeks about this one. But my question is, what happens in 6 months? It seems like every 6 months or something, we get an alpha, a delta, an Omicron, a really big thing. Are we going to be due for a new one? There's certainly lots of viruses out there that are evolving. So there might be another one. There might be another one in the works. And if things work out for us, our vaccines will continue to protect us against it. But if we sort of drop the ball on vaccine development, we could be looking at a new form of SARS-CoV-2 that our vaccines are just not really good against. So it's been a long 2 years, but I see no reason to just get complacent.
Gosh, sounds like we need to be conscious, and this pandemic could just ooze on its way out. And maybe, I guess well, let's not leave it on a pessimistic note. We might have this with us forever. But anyway, thank you so much, Carl, for taking the time to talk to us today. And thank you for all the wonderful work that you do, making the science, in particular during this pandemic, intelligible to the rest of us. So, so on behalf of the Frankie program and our audience today, I'd like to give you a big, warm thank you.
Thanks so much for having me. It's been a lot of fun.
And thanks everyone for attending. We have a couple of events coming up next. We are going to continue virtually on Zoom through to the end of this semester. And we have our next talk on the mapping series, which is coming up, which is by the musicologist Victor Coelho. And then we have 2 talks coming up by our own Frankie Fellows, Frankie Graduate Fellows, in April as well. So we have a rich program for next month. The Mapping as Knowing lecture series talk by Victor Coelho is on Tuesday, April 12th. And the talks by our Frankie Fellows, Ana Florence and Diego Ellis Soto, are on April 20th, and dates to be decided further later in April. Anyway, I would like to thank everyone for attending and look forward to seeing you at the next events. And thanks again, Carl. Bye.
Bye-bye.